Hello everybody. As you are all aware that we are talking about surface chemistry, the fifth chapter of your chemistry textbook, that is part 1. In the previous lecture we studied classification of colloids. In one of the classification criteria we studied two types of colloids, lyophilic and lyophobic. Clear? In that we discussed that lyophilic salts, they are easy to prepare, but lyophobic salts, they require some special techniques. So today we are going to talk about preparation of colloidal salt, but we will focus on lyophobic salt. Clear? So which different techniques, special techniques are used to prepare such salts? So the first technique is a chemical method. In chemical method, we don't have to go for any description, just the name of the method and the reaction involved in it. Only that much we have to remember. Generally, this is asked in 1 or 2 marks question, or it can also be asked in 3 marks question, as there you can see there are 4 different methods. Clear? So it can be asked in any form. So here in chemical methods, the first method is double decomposition. Example is arsenic oxide. When it is reacted with hydrogen sulfide and double decomposition is carried out, then arsenic oxide is converted into arsenic sulfide, and this sulfide is called salt. Clear? Second method is oxidation, in which sulfur dioxide is reacted with hydrogen sulfide, so sulfur salt is prepared. Third technique is the preparation of gold salt by reduction method. So here AuCl3 is reacted with formaldehyde and water, so we get the salt of gold, and formic acid and HCl are obtained. Fourth technique is hydrolysis. Here FeCl3, ferric chloride, when water is added to that, then there will be hydrolysis and the precipitates of FeOH thrice, means ferric hydroxide, will be obtained, and that is known as a salt. Clear? Now here, from MCQ point of view also, you can be asked that which of the following is not the chemical method for preparation of lyophobic salt. So 3 options may be given out of these 4, and the 4th option is something totally different. Clear? So from MCQ's point of view also, this topic is important. Now either we apply any of the 4 techniques, double decomposition, oxidation, reduction or hydrolysis. Whatever products are obtained, these products, they are then converted into colloidal particle size. They are then mixed with suitable dispersion medium, and colloids or the salts are prepared. Clear? Second method is electrical disintegration or Bredig arc method. This is asked in 3 marks or 2 marks question. Here diagram is compulsory. If it is asked in 3 marks question, then diagram will carry 1 mark. And if it is asked in 2 marks question, then diagram will carry half mark. Clear? So, and diagram is very simple to draw. So don't skip it. You have to draw this compulsorily. And this method is again very important from examination point of view. Electrical disintegration or Bredig arc method. The peculiarity or the characteristic of this method is it involves 2 simultaneous techniques. First is dispersion. First technique involved in this is dispersion, and second technique is condensation. So combination of these two will help us to prepare salt. First of all, which type of salts can be prepared by this technique? So gold, silver and platinum. If we want to prepare the salt of either of these three metals, then we have to go for electrical disintegration method. Now here in diagram you can see there are two containers. In one bathtub, two metal electrodes are immersed. These metal electrodes are either of gold, silver or platinum. Clear? So these metallic electrodes are immersed in a suitable dispersion medium. So this is the dispersion medium, this blue color. These two electrodes, a strong electrical arc is struck between these two electrodes, so that immense heat is produced by this electrical arc and metal will melt. Clear? And it will get vaporized, not exactly melting, but solid metal is directly converted into the vapor phase. So this metal will get vaporized. But as these metal electrodes are dipped in dispersion medium, vapor which is formed here from these metal electrodes, it will be condensed. Because you can see in the outer container, it is called ice bath. Here ice pieces are filled up. So that by vaporization of this metal electrode, the metal particles which are obtained, they are present in the dispersion medium, and outside this medium there is ice. So condensation will take place, and so these particles will be converted into the colloidal size particles, and that is how colloid of either gold, silver or platinum is obtained. Clear? So this method is the combination of two techniques. First is dispersion, when a strong electric arc is struck between two metallic electrodes which are immersed in the suitable dispersion medium. Then due to the heat produced by this electric arc, these metal electrodes will vaporize. But as this vapor cannot go out because the metal electrodes are immersed in the dispersion medium, and that medium is surrounded by ice. So as soon as metal vaporizes, the particles which are formed, they will be condensed to the solid particle, and that solid particle will be of colloidal size. Clear? So this is second technique. Third method is peptization. Peptization means converting freshly prepared precipitates into the colloidal size particles. That is called peptization. Here, in order to convert precipitates into colloidal particles, some suitable electrolytes are needed. Clear? This electrolyte is to be taken in a very small amount, so that the colloid which is formed will get stabilized. Clear? So this electrolyte which is taken in the small quantity is known as a peptizing agent. So for this technique, presence of peptizing agent is must. Here I told you that freshly prepared precipitates, they are converted into the colloidal particle size. For example, FeOH thrice. Here you can see by hydrolysis reaction, when FeCl3 is reacted with water, it gives precipitates of FeOH thrice. These fresh precipitates are taken, and into that a very small amount of electrolyte like FeCl3 is added, so that now this electrolyte has positive and negative ions. So this precipitate, it will absorb any of these ions of the electrolyte. If it absorbs positive ion, then these precipitates will become positively charged. And if the precipitates absorb negative ion, that is chloride ion, on its surface, then the precipitates will become negatively charged. Now, due to addition of peptizing agent, freshly prepared precipitates get converted into positively charged particle. It depends which ion of the electrolyte is getting absorbed on the surface of precipitate. So accordingly, it will get converted into positive or negatively charged particle. Now when these precipitates become electrically charged, then they will start breaking into the small particles, and these particles will be of colloidal size. We know in chemistry practicals, when we do inorganic analysis, then in the given solution, when we add some reagent and if precipitates are formed, then these precipitates usually they settle down at the bottom of the test tube, because these precipitates are not of colloidal particle size. Is it clear? But to this freshly prepared precipitate, if small quantity of suitable electrolyte is added, means peptizing agent is added, then positive or negative ion of this electrolyte will get absorbed on the surface of precipitate. So now this precipitate, they become electrically charged, and that charge will be identical. Either all precipitate particles will be positively charged or they will be negatively charged. Clear? So there will be repulsion. So they will start breaking into the smaller particles. Now these particles are of colloidal size, and that is why now this precipitates which were settled down, now they are converted into colloidal size, and that's why this colloidal particle will get now dispersed in the whole solution. Is it clear? So that is how colloidal sol is prepared. So peptization is a very important technique. Again, this can be asked in 2 marks question. Clear? So what is peptization? Peptization is the process of converting freshly prepared precipitates by addition of small amount of suitable electrolyte into the colloidal size particles. So that process is known as peptization, and the electrolyte is known as peptizing agent. Clear? So these are 3 important techniques which are used to prepare lyophobic sols. Clear? All of them are very important. Okay. Now once the colloid is prepared, once the sol is prepared, it is again necessary to purify it. Clear? Now why should we purify the colloidal sol? Because as in this technique we have seen that electrolyte is added to the freshly prepared precipitates. Now sometimes these electrolytes, they are present in excessive amount. So if electrolyte is present in a small amount, then it will stabilize the colloid. But if it is present in excessive amount, then it will coagulate the colloid. Means the colloidal particles will start coagulating, they will start aggregating and will start settling down at the bottom. So the colloid will now of no use. Is it clear? So the excessive amount of electrolyte or some other soluble impurities are present in the sol, then it is always necessary to purify the sol. So here, what is purification of sol? Purification of sol is to reduce the amount of impurity to certain requisite minimum. That process is known as purification. Again I repeat, purification of colloidal sol is the process of reducing the amount of impurities to a requisite minimum. Now these impurities can be excessive electrolyte or other soluble compound. Is it clear? Okay. Now there are 3 techniques to purify the colloid. First is dialysis. This is very simple technique. In this technique, this type of bag is taken. For example, suppose we have a polythene bag, and in that polythene bag if we put something, clear, and then we tie it with the thread or rubber band. So that type of arrangement is there. So in the bag, now this is not ordinary bag or the polythene bag. This bag is made up of a special type of membrane. This membrane will allow only the impurities to pass through it. Impurities means the ions. If it is the impurity of electrolyte, then the ions are there. And if some other soluble impurity is there, clear, then that is also to be removed or it is to be separated. So this membrane of the bag, it allows only the impurity to pass through it, but colloidal particles cannot pass through it. Clear? So generally this membrane is known as dialyzing membrane, and this dialyzing membrane is either animal membrane like the urinary bladder. Clear? The membrane of that urinary bladder is a semi-permeable membrane, or it is called the dialyzing membrane. Clear? Another is parchment paper or cellophane sheet. Clear? This is again important MCQ. So out of these three, we can use generally parchment paper or cellophane sheet is used. Animal bladder we cannot use for dialysis process. Clear? So it occurs that dialysis when some person has kidney problem, then artificial dialysis is carried out. Clear? When his kidney stops working, clear, it stops purifying the blood, then that fellow or that patient is put on artificial dialysis. Clear? So here this membrane, dialysing membrane of this bag is either parchment paper or cellophane sheet. Now in this bag we have to put our colloidal sole and then we tie the bag. Clear? Either by rubber band or thread, and then it is hung from some clamp or some stand. Then that bag is immersed in a bath which is filled with water. It is a pure water. So this is the inlet from where pure water continuously enters this container. Clear? And this is the outlet. Now as I told you that the membrane or the wall of this bag, the material of this bag, it will allow only the electrolyte or the soluble impurities to pass through it. So this electrolyte or soluble impurity will come out from the membrane or the wall of that bag and it will mix with water. From the outlet, water plus crystalloid. Crystalloid means the impurity. So that will come out and only colloidal sole particles will remain in the bag. So this is called dialysis and this apparatus is called dialyzer. Clear? But this is a slow technique. So in order to fasten it, we go for second method, that is electrodialysis. Diagram remains same. Only additional thing is here electrodes are there. Clear? This technique is more preferred when the impurity is only electrolyte. No other impurity is there. Only electrolyte, excessive electrolyte is present as an impurity. That electrolyte has positive and negative ions present in it. Clear? So in this bag, the colloidal sole containing excessive of electrolyte is filled up and that bag is immersed in the water bath. Clear? But here two metal electrodes are used, means electric supply is given. This electrode is called anode and it is connected with the positive terminal of external source. And this is cathode which is connected with negative terminal of external source. Now when electricity is passed, then what will happen? Electrolytic ions which are present in the colloidal sole, positive ions will be attracted towards cathode and negative ions will be attracted towards anode. So that is how fast dialysis will take place. Positive and negative ions of electrolyte will get separated very fast because electrical field is applied. Clear? And so they are attracted towards the oppositely charged electrodes. So this technique is fast. Got it? So here you can see in the diagram, here pure water, from the outlet water plus electrolyte will come out. Here I have written crystalloid. Crystalloid means it can be the soluble compound or the electrolyte, both together. Clear? But in electrolysis, this water which comes out from the outlet, it contains water and electrolyte. Clear? Okay. So this is another purification technique. Here I have written salt particles plus crystalloid. Crystalloid means impurity. Clear? Third technique is ultrafiltration. Ultrafiltration. Filtration word we have used. Clear? In chemistry lab we carry out filtration many times using ordinary filter paper. Clear? But that ordinary filter paper, they have larger pore size. So colloidal particles can pass through it and filtration cannot be carried out. Clear? So ordinary filter paper cannot be used for ultrafiltration technique. It means ultrafilter papers are used. Now what is this ultrafilter paper? In that, ordinary filter paper is impregnated by colloidal solution. Clear? It is impregnated means it is soaked in colloidal solution. Generally, this colloidal solution is a 4% nitrocellulose which is taken in the mixture of alcohol and ether. Clear? So mixture of alcohol and ether, into that we add 4% nitrocellulose. So this mixture is known as colloidal. Clear? Into this colloidal solution, ordinary filter paper is soaked. Then it is taken out and it is hardened with formaldehyde. Clear? HCHO, methanol, which is commonly known as formaldehyde or formalin. So this formaldehyde, in order to harden the filter paper, formaldehyde is used and then that filter paper is dried. So these are called ultrafilter paper. Now in ultrafilter paper, pore size is reduced compared to ordinary filter paper so that colloidal particles cannot pass through it. Clear? And then the filtration is carried out. Got it? So using filter paper, we will get the impurity. Clear? And colloidal particle cannot pass through the filter paper, so they will remain on the funnel. Is it clear? They will remain as a residue. Clear? Then using proper dispersion medium, again the stable colloidal salt can be prepared. But here again this ultrafiltration is quite slow. So in order to fasten it, sometimes we have to apply extra pressure or suction. Clear? So by applying pressure or suction, ultrafiltration can be faster. Is it clear? So these are three different techniques which are used for purification of soil. All of them are very important for 2 marks question. This can be asked in 1 mark MCQ, 1 mark objective type or 2 marks objective type question. Is it clear? So today we have discussed preparation of colloid and purification of colloid. In the next session we will go for properties of colloid. Till then, have a nice time. Thank you very much.